Okay, cool. So viruses, and this is actually a slide that I stole from Dr. Chrisman. I really do like it though, because it kind of gives you perspective of the size of viruses. And if we were to be in lab, would help you understand why, right? You can't see them underneath a light microscope. The lighter, I don't have it in here. I just have matches. So you're looking at the size of the virus, and what you see here, right? And I know some of you don't like baseball, but too bad. I do. And if you're kind of using the size of a baseball park, right, as your starting point, right, that would be the equivalent of a human cell, right? So if some of you, and I'm not talking about the Chattanooga Lookouts, right? Some of you have been maybe to Atlanta, right, to SunTrust, to their new stadium or some other place. That's what I'm talking about, right? Professional baseball park, holding somewhere from 30 to 70,000 people, depending on where you go, right? And so if that whole park is equivalent to a human cell, well, then a bacteria, right, is equivalent to the pitcher's mound, which you can see right down here. So you can still see that. But then a baseball, right, is about the equivalent to the size of a virus, right? So if I gave you had the view of the whole kind of park or stadium, right, and there was one baseball on the field, okay, you wouldn't be able to see it, right? It's super, super small, all right? To give you another kind of perspective, and this one works better if you guys would have been in lab and would have been able to see E. coli under the microscope. But mind you, E. coli is actually a relatively small, kind of short, nubby rod that is relatively hard to see. And here you can see even some of these larger viruses, all right, are still very small compared to the size of E. coli. And one of the other things we'll talk about here in just a second that you can probably also see is that viruses are coming in not only different sizes, but different shapes. Right. So we're not going to spend a lot of time here, but one of the things that's always interesting to think about is, as scientists or medical professionals and researchers, is where did viruses come from, right? Why are they here? Okay. And so one of the theories, right, is this regressive or degeneracy hypothesis, where these viruses were some type of former parasite, perhaps prokaryotic or even eukaryotic. And the idea being that maybe there was some type of extreme environmental stress, and this group of organisms kind of figured out what they could get rid of, what they didn't need, and basically went down. If you've heard of going down to the bare bones, all right. If you're one of those people that goes away on a weekend trip and you fill 2 large suitcases, and maybe your parents or your significant other are like, yikes, trim it down. And that's kind of the idea here, is that these organisms trimmed it down to basically figure out what's the minimum, right, proteins, enzymes, nucleic acids that we need in order to survive. Cellular origin hypothesis, vagrancy hypothesis also called. So that's the idea of we had some rogue plasmids from prokaryotic cells that left, right, broke away from the main bacteria and were able to survive on their own and eventually become to what we know as viruses. And then, if I had to pick one of these, this is probably the one that I settle on the most, is this idea of coevolution, all right? And so the fact that viruses have been present as long as life has been present, okay? And, well, yes. And there are all kind of interesting ways that you can, bring a Christian perspective into all of these, but we can talk about those via email, Zoom, or office hours in the fall when I'm sure you guys all come to visit me in person, right? So what can viruses infect? Plants, animals, bacteria, and perhaps even other viruses. So basically everything, right, that has some aspect of life associated with it. Now, the thing that's really cool about viruses, and it's always kind of one of those debates that I'm sure you hear people talk about, even if you've read stuff in, Rachel, where was your article from?
Mine was from Science Daily.
Science Daily. All right, so Science Daily would have stuff on this like this if you read stuff from Scientific America, right? So, legit science sources that maybe aren't like primary research papers, but you'll always hear this kind of, well, are viruses dead or alive? And the answer is yes, yes. All right. So when we think of dead things, right, you think of things that are crystallized. All right. And so sometimes in order to get protein structure, we crystallize proteins, and we actually see that some viruses are already crystallized, right? Inert, no cell components, no metabolism, no autonomy, right? So what does that mean? Well, that means, right, inert. So they're, I don't know if you stopped at the side of the road and saw a dead raccoon or possum and you poked it, right? What would it do? What would it do if you poked a dead raccoon or possum on the side of the road? Glory?
Well, it wouldn't do anything.
It wouldn't do anything, right? Because it's inert, right? It doesn't. It's not doing anything, right? Viruses, in a sense, are inert, right? They're not living. So no cell components, right? Viruses are not going to have. And when we say no cell components, right, we're specifically talking about like they're not going to have a nucleus. They're going to lack organelles, right? One of the things they do have, and we'll kind of mention this, or I'll try to do a better job of mentioning this, is they do have enzymes, right? They do have nucleic acids, so you can actually see that over here, right? They're made of proteins, right? So specifically when we say when no cell components, we're really referring to more like no organelles, all right? Or organelle equivalents. No metabolism, right? So again, if that virus is just sitting in a petri dish, right, it's inert. It's not capable of breaking down or building things, all right? No autonomy, right? Basically, a fancy way of saying it can't live by itself, okay? It has to live in its parents' basement, right? Like some of you. Which one? Yeah, probably you, Sarah. That's my guess too, right? No, I'm just kidding, right? So no autonomy. It can't live on its own. So then you come over here and you say, okay, alive. I mentioned nucleic acids. Well, you have nucleic acids and you're alive. You have proteins, all right? We'll see viruses have enzymes, right? They also have proteins that make up some of their protective coat that we'll talk about. We know that they're capable of mutating, all right? So, I mentioned earlier with contagion, we talked about the flu, and you're like, well, why doesn't the flu vaccine work? Well, why does the flu vaccine not work perfectly, or why do we have to get a new one every year? Well, that's because the virus mutates, right? Why have we not eradicated HIV? Well, HIV is mutated. It changes, all right? They can grow and multiply, right? They can make copies of themselves. All right? Now, again, you'll see here today, and hopefully you probably already knew this, right? They need a host, and they need help doing that. And they can react, all right? They know when a cell is stressed out or when it's not stressed out. They know when they're trying to be eliminated, and sometimes they'll hide and incorporate themselves in the nucleic acid of a host cell, all right? And so they have characteristics of both, right? They're kind of in this little bit of gray zone, okay? Yeah, it's just the reward thing. Yeah. Okay. So we'll talk about all of this stuff in a little bit more detail, but viruses are incredibly diverse when it comes to the nucleic acid that they're carrying and how they're packaged, all right? So viruses can have DNA. They can have RNA. They can have single-stranded DNA, single-stranded RNA, double-stranded DNA, and double-stranded RNA, all right? And then, right, with the single-stranded, double-stranded, they can have positive, negative strands, and we'll see a little diagram that shows both of that. So they can have the coding strand, they can have the non-coding strand. They can make the coding strand from the, right, they have incredible capabilities to manipulate and utilize nucleic acid, right? Now, viruses can be naked, all right, or they can be enveloped, right? And you'll see what that means in just a second. Please note, and I'll try to emphasize it right, a naked virus still has protection, right? Naked for a virus does not mean naked as it would for us, right? There's still a level of protection there. All right, so viruses, right, have some really kind of cool, interesting shapes. All right, so icosahedral here, you can kind of see it's kind of this rounded. This is for, I assume, like Amanda probably plays like Dungeons and Dragons, right? And so those dice playing games, yeah, just okay. You don't have to deny it, right? You're being recorded though, so we'll go back and look at this, right? But this cool multi-faced kind of a circular a structure, and so you see some different viruses here: adenovirus and hepatitis. You could also throw coronaviruses into this group, right? Helical. And so you basically think of an alpha helix, all right? And again, you'll see another picture with all these kind of next to each other. A great example of this is the tobacco mosaic virus, which is actually the first kind of virus that was identified that allow us to figure out that viruses, were a thing. And then you have complex shapes, which is my favorite, right? Combinations of both of these. And you see here, this is a T4 bacteriophage, and we've already kind of briefly mentioned those in transduction. But here you can see it kind of looks like it has an icosahedral component here, but then it also looks like it has a helical component here. And so again, you can have these things combined, and here are these cool little spikes or the legs of the lunar landers. And we'll talk more about spikes here in just a second. So here is, showing, or this is a diagram that's kind of showing these things stacked together. It's also giving you a little bit of a size comparison, right? Bacteriophages are bigger than a lot of the viruses that cause infections in us, all right? And so some of these viruses you're probably familiar with feline distemper. If you have a cat, this is one of the things they've been vaccinated for. Foot and mouth disease, if you have a toddler, or if you don't have a toddler and have a toddler someday and they go to daycare, this runs through daycares like a burning rapid fire. We had the pleasure of getting to experience this. Rhinovirus, right, if you've had a cold, this is one of the causes. Dengue fever, I hope you guys haven't had, but maybe if you've done mission work, that could be the case, right? And you could throw HIV as a polyhedral or a icosahedral virus along with corona. And please note, right, that structurally, you see there's different sizes here. Shapes are similar. Even if you have a similar shape, right, the nucleic acid could be completely different on the inside of these viruses. All right, so let's briefly talk about a naked versus an enveloped virus, right? So here you see they call the naked also a non-enveloped virus, and it's on the top. And what you see here is it has its nucleic acid, right, packaged inside, and then you see here a caspid coat made of protein, and sometimes also attached to this caspid coat, right, you'll have enzymes that may be important for viral function, potentially. And then you have the spikes, right? And here, whether you're naked or you're enveloped, you see that spikes are present on both, right? Does anyone have a guess, or does anyone learned about viruses before. What are the spikes for? What do you think the spikes are important for when it comes to a virus?
Do they help the virus adhere to the cell, like outside of the cell, and like?
Yes, perfect, right? So the spikes, right, are going to be what allows the virus to attach to its target, right? And spikes are different on different viruses, and so different viruses have areas, right, that they infect, right? We'll talk about this in just a second, or at least mention it, right? COVID, right, 19, or the virus that causes COVID-19, its spikes allow for the attachment of the virus to lung tissue and to GI tissue, right, gastrointestinal tissue. Okay, influenza also, right, is allowing attachment to respiratory tissue. Something like HIV, right? Its kind of main attachment points are actually going to be some of your lymphocytes. So the receptors that it, or the spikes that it contains, allow it to attach to cells of your immune system, right? So we see all kinds of cool things here. In some ways, right? These are similar to the fimbriae that we see in pathogenic bacteria, right? They serve as that attachment point, all right? So this protein coat, right, provides protection for the nucleic acid. It also contains enzymes that can be active in driving the viral cycle. Enveloped is really cool because it still has that caspid, right, that surrounds the nucleic acid. But then we also see, right, this envelope, all right, that contains proteins. It also will contain a lot of times phospholipids from a host cell along with its spikes, right? So it kind of has this extra layer of protection around that nucleic material, and kind of this unique envelope structure kind of drives its interaction with its target cell. And we'll go over that in just a second here too. All right, so naked or enveloped. And viruses, again, because this whole are they dead, are they alive, means that classification systems vary. One of the common ones that's used is the Baltimore classification system. And basically, what this is doing is it's classifying viruses based on the starting nucleic acid that they have. And so like class 1 starts with double-stranded DNA. You can see down here, class 7, right, has double-stranded DNA with reverse transcriptase, single-stranded RNA with reverse transcriptase. So all these kind of cool, unique starting features. One of the things that I always try to emphasize to my students, no matter what you are starting with, all viruses are trying to end with something very similar, right? Or the exact same thing, right? They all want to make mRNA. Noel, why does a virus want to make mRNA?
So it kind of like disrupts your protein synthesis, like of your own mRNA.
Okay, it can. So, a lot of times, yes, there can be, yeah. I'm trying to think. It's not quite what I'm looking for. You're not wrong, right? But how about this? In order for it to interrupt the synthesis of your proteins, what has to be made from this mRNA? What do you think has to be made?
Proteins.
Proteins, right? Specifically enzymes. And so what we see with a lot of, viruses is that there are enzymes that interrupt the host ability to do its job. Sometimes those are included as part of the caspid. Sometimes those enzymes, right, are encoded within this mRNA, right? Proteins is also right, because the proteins that are part of the caspid coat of the virus, right, are made by this mRNA. And so you basically need to make mRNA because you're making proteins and you're making enzymes and you're getting ready to make copies of yourself. All right. And so the basic principle of, well, we'll say the basic principle, right? We go DNA, RNA, protein. Viruses are kind of crazy, but eventually, right, and you can see there's different ways to do it. They get to mRNA before they make protein, right? They need to make protein. They want to make protein just like we do. Right? Now, like I said, this is very brief because I'm going to incorporate some more coronavirus, COVID-19 things into future lectures. So we're going to talk about vaccines. So I'm going to put some stuff about corona in there. We'll also talk about antibiotics, and to that I will add a small antiviral section. So we'll talk about at least one antiviral in particular. Maybe you've heard of it, Remdesivir. I think I'm saying that correctly. Was discovered at the greatest university in the land, the University of Alabama at Birmingham, right, my alma mater, and that's becoming one of the potential frontline drugs for treating COVID. So that'll be in there. And then COVID is fascinating from an epidemiological standpoint. And so we'll look at some case reports from the CDC. And one of the ones that we'll look at is prisons. And so interestingly enough, some of the areas that we see really bad COVID outbreaks are in prisons. Okay. So we'll kind of talk about that. But as far as coronaviruses, right, there are hundreds of coronaviruses. And in case you're wondering, and I haven't checked to see what the oldest one is, but I found a research paper. I did not read it. I'm sorry. I have a lot of other things to read, but it was actually looking at coronavirus infections in dogs in 1986. All right. So the reason that we haven't heard about them or talked about them much is because for the most part, they don't cause issues in us. And again, we'll talk about this a little bit more in the epidemiology. I got to see if I can find this video. It's an interesting video, right? So this idea that coronaviruses typically jump from animal reservoirs to humans, and we think that this occurred in the Wuhan wet markets. And I'll show you a video. You can see that in the Wuhan wet markets, they have a lot of exotic animals that they bring into close quarter with one another. A lot of cages stacked on a lot of cages. You have a bunch of people that are there. They slaughter and sell the animals there. And so there's a lot of opportunities for transmission. And so this is where we think the SARS epidemic started, or actually we linked it to a wet market in China, and now COVID, right? And so its official name is SARS-CoV-2, right? The first virus is SARS-CoV. In the Middle Eastern, MERS, Professor Perry, do you know, was MERS linked to a wet market? I don't know as much of its history. It's not quite talked about as much.
I don't know a lot about the history, but they have definitely linked it to camels.
Okay.
Now, how the camels get it, I'm not certain.
Yeah. So camels are one of the reservoirs that it actually talked about in some of the papers that I'm looking at. But anyway, so, SARS, although some of you may have been around then, or a lot of you were around then, you were really little. And SARS, I think, infected only about 8,000 people worldwide, but it did have a 10% mortality rate, so it was pretty scary. And China and Canada, actually, interestingly, were the ones that were most affected by it. Severe acute respiratory syndrome. Middle Eastern respiratory syndrome was in 2012. I don't know as much about it. I might try to find some more on it maybe for the epidemiology lecture. And then SARS-CoV-2, right, which we're calling COVID-19. And so here you see 3 viruses. As far as the hundreds of coronaviruses that are out there, only 7 that we know of so far have been able to show or have been known to cause infection in human that induces some type of illness. And of those 7, right, these are the 3 that we know are capable of causing severe illness. And so what's really interesting about SARS-CoV and SARS-CoV-2 is that SARS, for the most part, the original pandemic in 2002, that virus appears to be more lethal. But, right, the virus that we're dealing with right now appears to be able to cause infections at a much higher rate, right? So it's more easily transmitted, right? So kind of interesting things. But again, right, for the most part, the illness that's caused by coronaviruses is very similar to the common cold, right? And we actually see that even with COVID-19, the majority of people that are infected experience no symptoms to mild symptoms, all right? And so there's still a lot that we have to understand here. Now, again, I already mentioned this, right? SARS-CoV-2, we see about an 80% genetic, or it's identical to the SARS-CoV virus. So very, very similar. We'll mention this again, but it's an RNA virus, right? It has a single strand, so it has the plus strand or the positive strand of RNA. And where it attaches, right, where it attaches its spikes, it actually interacts with the angiotensin converting enzyme 2, right, ACE2. And angiotensin, we'll talk about this more in A&P dose, right? But your lungs are actually playing an important part in activating angiotensin, which has been shown to, or does affect blood pressure. And so what's really interesting though, for the virus is that the spikes actually have to interact with other proteins in the host. And I'm not going to claim to know what all these two do, but transmembrane protease serine 2 and/or lymphocyte antigen 6E. And so basically the spikes encounter these proteins, it activates the spikes and then allows the spikes to attach to these ACE2 receptors. And as I mentioned before, the ACE2 receptors are expressed in the lungs and the GI tract. And so one of the things that you may have heard about COVID-19, besides some of the respiratory issues, is that there are people that are drawing correlations that perhaps one of the first signs of a COVID infection may be some type of GI distress, right? And that's because the ACE2 receptor is expressed in the gastrointestinal system. So, right, that's how it's getting to your cells. And so the spikes, right, on COVID, the COVID-19 virus, are allowing it to attach to this receptor, ACE2, which we find primarily in the lungs and GI tract. And I'm sure you guys have seen the picture of SARS-CoV-2 or COVID-19, the COVID-19 virus. And here you can see its spikes kind of look like little crowns, all right? And so all coronaviruses have this kind of general structure. And here you can actually see an electron microscopy of a cell that's infected with a coronavirus. And so this is a cross-section of the cell. So these viruses have basically been kind of chopped in half. And so you can see the little black dots on these viruses, right? That's going to be some of the spikes. And so you can see, compared to a cell, these viruses are really small. And I'm sorry I didn't give you like a size comparison here, but remember, electron microscopy is much more powerful than your light microscope, right? So this image is magnified significantly. And what's really cool, and you can't really tell here, but I'll show you a little diagram here in just a second, is the our coronaviruses like to hang out and kind of be assembled and do their thing in the endoplasmic reticulum and the Golgi of cells is where we kind of find them clustered together. And just really quickly, right, because these components of in this case the envelope of COVID-19 are really important in the infection cycle. And so here you can see the S protein, right? So this is the main spike. So the S protein is going to be what eventually attaches to that ACE2 receptor, right? It gives it its specificity. But you also see here present on the surface M protein and E protein. There's another protein that I haven't talked about, also called the N protein, that's on the surface. And so as I mentioned before, this S protein is what gives it its crown-like appearance, right? And gives it its tissue specificity. What's really cool, or one of the things that's cool, is these E proteins and these M proteins. And the M protein, right, they just highlight it here, and they don't do maybe as good as a job as I would have liked. This is from the CDC, so you can yell at them, right? But the M protein is the most abundant protein in that outer coat of the virus, right? That enveloped a virus, and it's going to be important in directing viron assembly. So a viron is going to be an individual viral particle that's being assembled in the cell. So that protein is important for that. And then this E protein, this is really cool, and I'll show you a picture of this in just a second, induces membrane curvature and scission, right, for budding within the Golgi, right? So what we see with viruses, and you'll actually see this in another diagram a little bit later, is as a virus gets ready to leave a cell, if it's going to be enveloped, a lot of times what we'll see is it'll go kind of to the cytoplasmic membrane. It has proteins that help it attach there, and then it kind of pulls a section of the cytoplasmic membrane off before it leaves. What we see with coronaviruses is that this E protein actually drives budding within the ER and the Golgi, all right? So they're fully assembled with their envelope surrounding their caspid before they even leave the cell, right? And so their assembly is actually taking place in your Golgi. And so here, right, you see this is the E protein right here. These green ones are actually M protein. And so this E protein, right, is inducing this bending, right, kind of getting that membrane to take the shape that it needs. And then what's really cool here is when you pull two E proteins together, that actually causes it to bud off, or right, to cut off. And then you have your assembled viral particle. And then you see here the red is representing the S protein, right, those spikes. And so that's just, again, that's just a little bit, we are going to talk more about, of course, symptoms and some of that other stuff, but I just want to kind of give you a little bit of that general information on the virus, right? So this is kind of there's some other viruses that do this, right? But this is definitely a unique feature, right? This is not common in a lot of viral infections, right? So again, that'll kind of be interlaced in our discussions for the rest of the semester. But let's go to one that you guys know a little bit about. We'll talk about this briefly, and then we'll take a little break and we'll come back. But bacteriophages, right? We've already mentioned these in the form of transduction. And so these are viruses that infect bacteria. It turns out that you have three kind of variations here. You have bacteriophages that are lytic, right? So think lytic lice, right? They cause the host cell to essentially burst open at the end of the infection cycle. Temperate, these are bacteriophages that are capable of lytic or lysogenic cycle. And you'll see what that looks like here in just a second. And then filamentous, all right, bacteriophages, you can kind of see they cause a unique infection cycle that we'll go over here in just a second. So what I'm going to do now is it's 9:12-ish. Let's come back at like 9:25. So I'll get you between 10 and 15 minutes, 9:25. And we'll start with the lytic cycle, and we'll work through bacteriophages, and then we'll see how much farther we can get in viruses for today, okay? And I know I have to add Mercy to the list. I remember she emailed me, so I'll add her to the list. And if any of you think of anything on break, you can go ahead and shoot me an email, and I'll go ahead and add you, all right? So 9:25, be back here and we'll keep on talking about viruses. There we go. All right. So let's talk about the lytic cycle of a bacteriophage. Right. So hopefully what you see here is something that reminds you of transduction, right? And it would be possible for a transducing particle to be produced, right, during this very thing that we're looking at. So as you guys are hopefully familiar with, right, we have attachment of the bacteriophage. Now please note, and we'll see some different things in animal viruses here, but the actual caspid, right, that protective protein coat along with the spikes that have allowed attachment are not entering the cell with the nucleic acid. All right, so here the nucleic acid is that cool neon green. See infection, right? When the when the phage DNA is injected into the bacteria cell, it results in the lysing and breaking down of the host chromosome, right? The cellular machinery, right, is then used to make copies, all right, to turn that DNA into RNA, and turn that RNA into protein, and we start to assemble new virons or viral particles. Right? So you make these different components, you then assemble it, and you have a fully functional phage, right? So it would be at this point when we're assembling our phage, if we were talking about transduction, right? Some of that broken down or segmented host chromosome would accidentally be packaged instead. Eventually you get so many phages that the cell lyses, you release these new virons, right? These individual viral particles, and they go and find a target cell. And here you can see an electron microscopy. I can't remember, this might be E. coli. I have to go back and check my source and see what cell this is, but you can see the bacteriophages attached to the outside of that bacteria. All right. So here is the lytic cycle. Notice, all right, that the lytic cycle has basically, I'm sorry, what am I doing here? Yes, I'm like, why am I doing that? The temperate cycle has two possibilities, right? So the temperate cycle, or a phage that is a temperate phage, right? All right, so you could be a lytic phage and not be a temperate phage. So just kind of keep that in mind. But a temperate phage has two routes it can go. Now, interestingly, no matter which way they go, it all eventually results in a lytic cycle, right, or a lysogenic phase. But we can immediately enter the lytic cycle, right? So this would be the same thing that we just talked about. Or, right, we can go into a lysogenic infection or a lysogenic phase. Now, truth be told, some of the thing that drives, right? So you're saying, I hope one of your questions would be, why would you choose one or the other? And sometimes it's completely random, right? So again, that randomness helps ensure the survival of the virus. And so if you have some of the bacteriophages that are temperate, they infect and they immediately induce the lytic cycle, where some go into the lysogenic cycle, then you're kind of covering all of your ground or your basis there, all right? So that's some of it. Some of it is also how quickly or how slowly bacteria are dividing. And so in some cases, the bacteria is dividing really slow. You might want to actually just go in the lytic cycle, make as many copies of yourself as you can, and then get out of there. If a bacteria is dividing really quickly, well, you can incorporate yourself into the host chromosome and you can get a lot of copies of yourself relatively quickly that way. And then when cell division slows down or the cell becomes stressed because of environmental pressures, you leave the lysogenic cycle, go back in the lytic cycle, make as many copies of yourself as you can before that cell dies, and boom. All right. So let's talk more about the lysogenic cycle there. All right. So in this case, right, in the lysogenic cycle, instead of chopping up the host chromosome, that viral DNA is integrated into the host chromosome. All right. So as I just kind of mentioned, as the cell divides, right, because bacteria cells divide as long as there's plenty of nutrients around, as that cell divides, it automatically makes a copy, right? The DNA polymerase that's making a copy of that chromosome does not know that it's making a copy of the viral chromosome that's integrated into that bacteria, right? So cell division, so every time, right, you have a generation here, right? So you have these two daughter cells, and then maybe this cell divides, maybe this cell divides, you're getting copies of that prophage. Now again, at some point it will leave. And some of this can be dependent on, is the cell dividing really slowly? Is it stressed out? And sometimes it will just leave randomly. Again, that randomness ensures survival of that virus. All right. So at some point, the phage DNA is excised, right? It leaves. And then just like before, when we talked about in the lytic cycle on the last slide, and you can see here, the host chromosome is chopped up, right? The phage DNA takes over the cellular machinery, makes copies of all the individual components of a viron, gets packaged, the cell lyse, and boom. All right. What's really cool, and we'll get to this, is that we actually see something kind of similar in HIV. HIV is great. So one of the things that makes it so hard to treat is it can actually integrate into a host chromosome and kind of hide out in your cells. And so every time one of your immune cells is activated, if HIV has integrated itself into a chromosome, a copy of that virus is made. So this is a really smart adaptation that some viruses have of this host chromosome integration. One of the things that's really interesting about bacteriophages is this idea of lysogenic conversion. All right. And so we have a couple of different things that happen. So if you see this term lysogeny, one of the things that's important for, right, is that it actually protects that bacteria from further infection. And so one of the things, if you go back and you look in your fungi unit, that's really fascinating about fungi is that when those hyphae are expanding towards their nutrient source, they actually secrete enzymes that repel other hyphae, right? And this is really important because you don't want to focus in on one nutrient source, right? So if we're talking about fungi, when one finds a nutrient source, it repels other hyphae, so those hyphae go a different direction and find a different nutrient source. And it ensures, right, that you're not relying on that one nutrient source, because if you eat up all of your hyphae from a fungi are focused on one nutrient source and they finish it off, well, that could be the end of the fungi, right? And so you kind of want to spread and conquer. And a little bit of a similar idea here. So lysogeny actually essentially sends a signal out to other bacteriophages. It repels them from infecting that cell. It's basically saying, hey, this cell's already infected. We're working it, right? It's good. We even see, although the mechanism is different, right? If we talk about fertilization of a female egg, right? Once that one sperm enters, right, it actually becomes impossible for other sperm to enter, and that's actually for the good of the cell. You don't need extra nucleic information in there. In this case, we don't need to spend extra resources on a cell that's already infected. Now, one of the things, right, that can result from lysogeny that can be kind of bad is sometimes, right, that the genes or the information that's encoded within the host chromosome is actually what gives bacteria their pathogenic capabilities. And so interestingly, you see these bacteria on the left, and some of them you've probably heard of, or if you at least heard of maybe the product on the right. And so, cholera, well, that'd be a good one to do a report on. Cholera is pretty interesting. But you see diphtheria, right? And so one of the things that you're vaccinated for, if you've had your DTaP vaccine, is the diphtheria toxin. I haven't checked, but I assume it's made a comeback, just like everything else that was pretty much eliminated in the 1950s because of anti-vaccination. But I'll get on my soapbox on that later. Botulism, right? You've heard of some versions of E. coli being bad. Truth be told, most E. coli is relatively harmless. But this is one of the really pathogenic strains that we get worried about. And so the idea here is that some of the pathogenic components of these bacteria are actually because of previous interactions at some point in the life of the bacteria, or in the evolutionary history of the bacteria, where they made friends, right, with a bacteriophage that incorporated genes into them that induced upon them pathogenic capabilities. All right. And so it's not that these bacteria were necessarily bad before, but it's actually after this interaction with bacteriophages that they have been given pathogenic genes and capabilities. Okay, so kind of a cool little thing. This is botulism, and I actually had Rachel, she sent me like this little article on it. And dented cans are okay as long as the seal of the can is not broken. I don't know how confident you are that that's the case. And there's something in there, Rachel, I can't remember. Do you remember about like you have to like thaw cans in the refrigerator if they've been frozen? I can't remember.
I think it was just like don't freeze cans or something, because it can.
I'm sure you could freeze them, you just can't thaw them at room temperature. I don't know. But anyway, right, for the most part, our canning process now is safe, right? But before, right, Clostridium botulinum, it loved these kind of nice anaerobic conditions. And so when we'd can, right, you take out most of the air, and boom, you'd open it, you'd eat it, and this toxin could kill you, right, because it causes a lot of muscle issues. So here you can see someone that has has botulism. They're not able, right, to control their muscles. But now, right, we use it for cosmetics. So if you've heard of Botox, right, that's injection of the botulinum toxin in specific areas. And again, this is not as bad. I'm not saying it's not as bad. It depends how you look at life, right? But you're not injecting the bacteria, you're just injecting the toxin. And so it's not like it's a full systemic exposure. And I just used, not that I want to pick on her, but, this is Demi Moore at age 30, and this is Demi Moore at age 46. I can guarantee you my progression will not be as good. And one of the reasons not to get on Hollywood celebrities, right, is but they're able to use things like Botox to hold on to certain things. Botox, interestingly, is also being used now for migraine treatments. I haven't really looked into that. I'll have to look into that and see if I can tell you more of the mechanism there. This, unfortunately, I haven't found a good link. I need to search again. What's been really interesting as global warming occurs is that it's actually led to the discovery of organisms that we did not know existed. And some of the organisms that we've discovered are actually giant viruses. And these giant viruses have simpler genomes, and also some of them are big enough that you can actually see them with a light microscope. okay? And scientists have actually been essentially reviving these giant viruses. But so far, the giant virus targets have been protozoa, right? So these used to be predators of simple eukaryotic cells, right? And so it doesn't appear that they have the ability to infect humans, but that's how most zombie movies start. So just beware, okay? But you can go and look at that. I'm not going to spend too much time on that. All right, so we've talked about bacteria phages. We're going to focus here a little bit on the filamentous bacteriophage, which interestingly, right, requires the presence of a fertility pilus of a bacteria. All right, so that means that it's going after bacteria that are capable of what type of horizontal gene transfer?
Conjugation.
Conjugation, all right? And so this is really cool. Let's see. No, go back. This is really cool. Let's see. I'm gonna show. I'll make sure I get this right. Yeah, that's what I thought. I'll make sure I said that right. Because this infection is what we call a productive infection. And if you notice at the end of this little diagram here, you see that the cell is not bursting open or is not lysed. Okay. And so this infection actually keeps the cell alive. So here you have your filamentous phage. It finds, right, a fertility pilus of a cell that's capable of conjugation, attaches to that fertility pilus, right, injects its phage DNA. And you can see here, right, that host chromosome is left alone, but you see copies of that phage DNA start to appear. All right. Now, productive, right? The cell's not dying. That bacteria cell is not going to divide as quickly as it used to because now the cell is essentially working double time, right? It's taking care of the bacterial chromosome along with the phage chromosome or the phage, not the phage chromosome, the phage nucleic acid, all right? And so you're making copies of both. But again, as the cell divides, it takes some of that phage DNA with it. And so, you know, the cells divide, the phage DNA goes with the new cell. It can then leave via the fertility pilus, go out and find another cell. Here it's kind of cool too. You see the capsomeres. And if you see capsomeres, I'm sorry I didn't mention this, but these are the individual components of the caspid. And here you see them lining the membrane of the bacteria. And so basically they fill that fertility pilus, go around, and it leaves and it pulls those caspid proteins with it so that it's fully covered and it's ready to go as it exits the fertility pilus, right? So kind of a cool infection, right? So this is productive because the cell is not dying. It's dividing much more slowly, but as long as the cell is alive and doing well, the phage is going to be present. I'm not really actually going to focus on generalized versus specialized horizontal gene transfer. If you want to go in your textbook and take a look at those, that's fine, but I promise I'm not going to give a question on those. But basically, you know, what that is referring to is in the cases where a bacterial DNA is packaged into the viral particle, there's kind of different variations of that, and that's what generalized and specialized is focused on. But don't worry about those. I'm not gonna go after those for you. Now, bacterial defense, and so you know one of the things that we're going to get into here soon is this idea of innate immunity and adaptive immunity. And that is your body's ability to protect itself on multiple fronts from viral, bacterial, protozoal infections, all of these pathogens. And bacteria, right, are no different, right? They have things that are trying to destroy them. A lot of times it's us. And so they have ways to defend themselves from different organisms. And so some of the kind of the basics, basic things that bacteria are doing, right, is that some of them are actually blocking receptors. And so if you remember when we talk about viruses, right, well, viruses are using their spikes to attach to cells, and a lot of times that attachment point is a specific receptor, sorry. And so if you block that receptor on your surface, well, then the virus can't attach to it. And so not all bacteria do this, but some do. And Staph aureus is a great example of a bacteria that does this, right? It produces something called protein A. Protein A is immediately secreted and then binds to the receptors on the cytoplasm of Staph aureus, preventing the binding of bacteriophages via that receptor. We've also talked about EPS, right? Slime and capsule, even specifically in the form of a biofilm, right? That's a sticky, sugary, mucousy-like substance. And bacteriophages can get stuck in there. And as long as you keep the bacteriophage away from interacting with the cell membrane, then you're going to avoid an infection. Interestingly, this slime and capsule can also be used to protect them from interacting with our cells that are trying to eliminate them, right? So it's a very good protective mechanism. What's really cool is that bacteria also produce restriction enzymes. So those of you that have maybe done genetics already, if you are a PA major, or if you're just a bio major, or those of you that maybe did some basic, I don't know, genetic experiments with E. coli in high school, have probably used restriction enzymes. And so restriction enzymes are enzymes that cut out specific sequences of DNA. And so in a laboratory setting, it's really cool because you can take a plasmid from bacteria and you can add a restriction enzyme to it. And so you know where it cuts. And because you know where it cuts, you can then take a segment of DNA that embedded within it is a gene that you're interested in, and you can insert it back into that plasmid, and you can get the bacteria to make a bunch of copies of your gene within that plasmid. All right. And so that's a really powerful research tool, but right, these restriction enzymes are actually used by bacteria to protect themselves. And so when they are infected by a bacteriophage, these restriction enzymes identify that DNA as being foreign, and they chop it up. So why is that potentially a problem, Anna? If I have an enzyme that chops up DNA as it enters the cell, why could that be a problem for the cell?
Could it chop up the wrong DNA?
It could chop up the wrong DNA, right? And that would be bad. So you don't want this protective enzyme chopping up your own chromosome, right? And so this is where bacteria have something called modification enzymes, right? And these modification enzymes, one of the things they do is they methylate DNA. And so if you actually go back, right, and remember our chapter on DNA repair, right, when we are looking for mismatch pairs and we're excising them and then replacing them with the correct pair, the way that we kind of know who's wrong, right, who's the new strand and who's the template strand, is that template strand's been around for a long time, so it's methylated. All right, so that means we replace the non-methylated segment where we see the mistake. The same basic premise here. Viral DNA that enters a cell has not been there for very long, so it has not been methylated yet. And so these enzymes, right, are able to look at it and say, hey, you just came here, you're brand new, you're not part of the cell, I'm going to chop you up. All right, so kind of a cool defensive mechanism that they have. Now, CRISPR, and some of you, if you haven't found an article yet, this could actually be kind of an interesting kind of twist or loophole in microbiology. So CRISPR is actually one of the main mechanisms that we're using now or that we're trying to use for genomic therapy. All right. And so we're actually using this in animals to change genes. But CRISPR is actually essentially the equivalent of a bacterial immune system. And so what you see here, right, is that the bacteriophage attaches to its target, inserts its phage DNA. The bacteria has something called a Cas protein that intercepts that phage. It chops up that phage, right? Kills it, just like the phage would want to do with the host chromosome, right? This Cas protein does to that phage DNA. And then what's really cool, right, is that little chopped up piece, or one of those chopped up pieces, actually inserts itself into a chromosome in something called a CRISPR array. And so here you can see it showing this phage DNA as green. So here's the green here. But here you can see these other colors in the array. And what these other colors are representing is previous infections. All right. And so in a kind of cool way, this is similar to the idea of immunologic memory, right? So immunologic memory is something that protects you from getting the same disease twice, right? Or this is the basic principle of vaccines, right? You go in, you get a vaccine, you create immunologic memory, and you're protected from future infections. And so the same basic premise here. So the bacteria essentially is keeping an infection history, all right? And so because this Cas protein intercepted, it avoids a viral infection. This has been inserted into its memory. What it starts to do now is when the when the CRISPR array is transcribed, it basically leads to these little, it calls them CRISPR RNAs. They're basically little identification sequences that get loaded into the Cas protein. And so when that infection is seen again in the future, that Cas protein is able to immediately recognize it, destroy it, and neutralize it before it can cause any type of infection, right? And you would see here that this bacteria has been infected by multiple bacteriophages. It's also a cool way to take a medical history of a bacteria because you can look at all the infections that have occurred, right? But we are now utilizing this mechanism for gene editing, right? So this is kind of, I would say, the present and likely the future, at least for a while, of gene therapy. Okay, and it's proving to be effective. I can't remember. Let me see. Hey, Professor Perry, do you remember? Was the gene editing that was done in China on those babies, that was done with CRISPR, wasn't it, by that scientist? Do you recall?
No, which I'm not sure what you're talking about.
Okay, so there was, it happened within the last year, and this Chinese scientist, I think, has been removed, but they knew that there would be a genetic defect in some children that a mom was, carrying, and so they actually used CRISPR to edit out the mutation, and the babies were born normal and healthy, which is fascinating, but is also highly unethical at this point. So, right, this is kind of the future of gene therapy, but before it was gene therapy, right, this is actually a bacteria's ability to deal with infections and to keep an infection history. Right?
So did that doctor or like researcher, did he do it in like utero or when they were born? Like,
so that's what's no, so it wasn't when they were born. And what I would have to check is, I want to say they were born via IVF, right, in vitro fertilization. And so I think he edited the embryos before they were implanted. I'll go check. Maybe if some of you don't have an article yet, or have an article that you're like, this isn't that interesting, someone see if they can find that one. I don't know if I'll get to it, but I believe he used CRISPR, so it's kind of cool. All right, so animal viruses, right? What we focused on primarily here was bacteriophages. Now I did talk about SARS-CoV-2, right, or the COVID-19 virus, which is an animal virus. And we'll see that there are a lot of similarities, but there are a couple of differences. If you don't know who this is, right, you're wrong, and you need to look up Animal on the Muppets and maybe spend your time watching some classics in between studying. Okay, Noelle, do you know who this is? Did you know who that was?
I didn't really watch the Muppets, no.
A lost generation. All right. So we're going to talk about an animal virus infection cycle, and you'll see that, again, there is a lot of similarities between the infection cycle of an animal virus and a bacteriophage. And so we'll talk about attachment and absorption, genomic entry, which is an important point that we see some variations, synthesis, assembly, and release, especially of these enveloped viruses, is unique in this case. And so here you see two types of entry into the cell, right? These are both enveloped viruses. Your first entry option is via membrane fusion, and then your second option is via endocytosis. And so you see here, they look really similar. Here in membrane fusion, you can see the spikes find their target receptors, all right? They bind to those receptors, and they cause the membrane to open up, and it's the nucleate caspid, right? So it's the caspid, right, with the nucleic acid inside of it that enters the cell, right? So it does not bring its envelope. It does not bring its envelope in with it, all right? Your caspid then breaks down, all right? And sometimes that will have enzymes in it that are helping you nucleic acids expose, and then you can start making copies of that nucleic acid and start making mRNA so you can make protein. Here, endocytosis, in this case, you start off very similar, but right, the attachment to these receptors actually triggers endocytosis. So you engulf the whole virus, right, the caspid along with the envelope. But again, the same basic premise. You're eventually going to break open that caspid so that you can expose the nucleic acid, make copies of it, make mRNA out of it, and start making new viral particles. Okay. Now, this is just a diagram. Again, I'm not going to expect you to memorize this, but I had mentioned, we looked at that Baltimore classification system. We're classifying bacteria like 1 through 7. So, essentially how are bacteria starting? This is showing you some of these different possibilities. So here you have DNA viruses, double-stranded. There we go. Sorry, the Zoom thing was blocking me. So you have double-stranded DNA, right? Single-stranded DNA. And in this case, right, they start a little different. So that's double-stranded DNA, you make mRNA from it, just like we would before, right? So there's that single-stranded positive strand RNA, the mRNA, and you can take that and directly make protein, all right? Here, single-stranded DNA, in order to interact with the cell's machinery, you have to make it double-stranded. You can then get your single-stranded RNA, you can then get your protein. And here you see different options. Again, highlighting, what do you know? At the end of the game, they're always having positive strand, right, RNA that is becoming protein, no matter where you start, okay? And so here with COVID, right, you're starting with actually single strand positive RNA. And so you can make copies of that to put in the new viruses by making it into negative strand RNA and then making it into more positive strand RNA, or you can take your positive strand right, you're doing both of these things at the same time, using that positive strand RNA to immediately make protein, right? So this is where the COVID-19 virus would be here. But again, no matter what, right, the end game is always protein, no matter what you're starting with. And here down here's this interesting, right? You have reverse transcriptase, which is starting with single strand negative DNA, and it's using reverse transcriptase to actually turn it into double strand RNA. So this is something called a retrovirus. This is how HIV works. Okay. Now, assembly, right? So basically, when you've made enough individual viral components, you can start to put a virus together again. And so you make that caspid, you put your genome in it, you start to put together everything else, and then you're good to go. What's really cool is that you can actually modify this viral genome as it's still in the host cell and as it's leaving. And so again, one of these aspects of this being alive, right? The viral genome can be changed or altered. And what's really cool here is I have spontaneous. We don't really have a great understanding of how viruses know this, but when they reach that critical threshold of having enough components around to start assembling, they just start doing it, right? And so there still is a lot to know. Now, for release, here you can see, like, let's say you put together your caspid, your nucleic acids in it. You have viral proteins that attach to the cell membrane, right? That help get the cell membrane ready for this virus, right? The virus then starts to cover itself with the host membrane, and it buds off, and here you have your enveloped virus. So remember, most enveloped viruses, large components of that envelope are going to be parts of the host cell. I also like this diagram because this is a little different than what's happening in the COVID-19 virus, right? The COVID-19 virus, right, this formation actually takes place inside the cell because the budding is occurring in the endoplasmic reticulum and the Golgi. And so for COVID-19, you'd assemble all of these inside the cell via the Golgi and endoplasmic reticulum, and that cell would eventually lyse and burst. All right, so a little bit different. I'm not going to take the time to click on this, but we focused on enveloped viruses. And what's cool about non-enveloped viruses is non-enveloped viruses, one of the things they do is they induce something called apoptosis. And so they actually don't interact with the host membrane because non-enveloped viruses don't need this component, right? It's not part of them. So they induce apoptosis, right? Or the good ones induce apoptosis, all right? So apoptosis is natural cell death. Why would you want to induce apoptosis if you were a virus rather than just lysing a cell open? Caroline, what do you think?
So that it doesn't kill the bacteria.
Well,
The
right.
cell?
So now, right, we're talking about animal cells, so there's no more bacteria, right? This is in your cell. All right. So why is apoptosis a good thing? Can someone help her out?
If there was like something wrong with like a cell, like it was like growing like uncontrollably, and then it like didn't want to like keep multiplying into being like something wrong.
Okay, well, so I mean, okay, yeah. So your body uses apoptosis as a relatively normal function, right? You want cells to die, right? Rachel's right. I think you want cells to die because as cells live, right, mistakes start to happen. You increase your chances of something like cancer potentially occurring. If any of you are into developmental biology, right, specifically embryology, apoptosis is a normal part of development. When you guys were being developed inside mommy's tummy. All right. And those of you that are A&P gurus, right, we'd actually say inside mommy's uterus, but that's fine, right? Those of you that don't know that yet, I don't want to ruin the lie for you, right? So we'll still say belly, right? But as your body is growing, one of the things that also happens is you have events of massive cell die-off, which is totally normal, right? Programmed cell death. Some cells were useful in development and helping get things started, but you don't need them anymore, and it's totally normal to happen. And because it's normal, one of the things that doesn't happen is your immune system is not triggered, right? Your immune system gets typically more hyped up for something called necrosis, right? So necrosis is when a bacteria or a virus causes cell death, right? And it's kind of, if you heard like necrotic tissue, think of necrotic tissue, right? It's kind of nasty, it's smelly, it's pussy, it's drainy, it's fluidy, right? It's an attention grabber, right? Necrosis is an attention grabber for the immune system. And so some viruses are really, really smart because when they induce apoptosis, it takes a lot longer for the immune system to realize something's wrong, okay? Because it is basically, the video that I show here is if any of you have seen demolition of big buildings inside of the city, right? They can't blow them up and cause them to scatter, but you can actually place charges that cause the building to essentially fall in on itself without causing damage to the surrounding buildings. That's what this video is showing, demolition in this way. And that's kind of what apoptosis is. It doesn't draw near as much tension and doesn't trigger tissue that's in the area. All right. One of the other things to talk about viruses, and we'll talk about, and some of you guys will actually talk about these in your presentations, is different viral infection cycles. And so you have acute, and then you have persistent. So acute, here it's using influenza as an example. At the moment, we would put the COVID-19 virus in this same category. So basically, there's a time when the virus is assembling itself, making copies of itself. That's kind of this peach color here. The orange is when you actually have the disease, you're showing a lot of symptoms. And then even after you stop showing symptoms, the virus is still present. So you're still infectious, but your body's dealing with it well, right? One of the things that I'm sure you've heard of that's so challenging with COVID-19 is that a lot of patients actually just kind of stay or are in this infectious phase for a long time and either don't enter a phase where they show symptoms, or the symptoms that they show are very mild. And so, that's one of the big things. If you hear about, if you've seen the masks carry, like the people that are saying, hey, wear a mask, wear a mask. A lot of times, it's not necessarily to protect you, although it's been shown to protect you a little bit. But where we want the most benefit is if you're one of those asymptomatic carriers, right? You're one of those people that has the virus in you, but never really enters strongly into this symptom phase. The idea is to keep you from breathing on other people, all right? So a chronic infection, an example of that would be if you've heard of hepatitis viruses. So, hepatitis B, there's also hepatitis A and hepatitis C. But in this case, what happens is you get infected, you have a short period where you show a bunch of symptoms, but then the symptoms go away, but the virus remains ever present in your system and in high quantities, okay? Now, when we start to talk about hepatitis viruses, please do not think that these patients are fine. While that virus is present, it is causing severe damage to their liver, but this is kind of, dangerous because if you're not tested and you don't know you have it, you kind of get over this initial bout of illness. You think you're fine. You interact with people in various ways, not knowing that you have the virus, and then the virus can easily be spread. Most of you have hopefully been vaccinated for Hep B, though. And then I think one of the really kind of interesting ones here, right, is I'm sorry, and chronic and latent are both in persistent, right? But this latent infection, so those of you that have cold sores, right, and unfortunately, right, a lot of times we think of genital herpes, right, is a form of a cold sore, but a lot of folks have cold sores. It's common to have those if you ever get the cold sores around your mouth, or cold sores actually infect the epithelial tissue, or the virus can infect the epithelial tissue inside your mouth, right? That is not an indication of a sexually transmitted disease, or as we're calling them now, sexually transmitted infections. And so these can be, relatively common. So those of you that decide to have children, if you have cold sores, one of the things they tell you is, especially during a flare-up, to be really careful to kiss when you're kissing your baby, right? Because you could kiss your baby, and right, if you kiss your baby near the mouth, you could spread those cold sores to them, right? So, one thing is it can be a sexually transmitted infection, but it's not always a sexually transmitted infection, right? You could also throw the chickenpox virus in here, but basically what's really fascinating about latent infections is that, this idea of flare-ups. And so here you can see a flare-up. It resembles an acute infection. It even appears to go away like an acute infection. And unlike the chronic infection, during this time, right, or a large chunk of time when you're not showing symptoms, you actually aren't infectious, right? So, this is a huge problem when we do talk about cold sores as an STI. Because you start to feel better, right? Maybe you engage in sexual intercourse and you think you're in this part of the non-infectious, or you think you're in this part where you're non-infectious, but you're actually over here, and then you spread cold sores to whoever your partner is. And so then you have a flare-up. Again, this can be caused by a lot of things. We're talking about cold sores on your mouth. Stress can be one of them. If you decide to have a child, sometimes the hormones and stresses associated with pregnancy cause kind of constant flare-ups of cold sores. There's a lot of different things that happen here. So, that's why those of you that have had chickenpox, although I think a lot of you have probably been vaccinated, the chickenpox vaccine was not even a thing when I was a kid, and I'm sure Professor Perry could still also speak, because it was the same way when we were both kids, even though our age difference is a few years, but during my time and her time, it was not uncommon to have pox parties, right? And so one kid has it, and another parent is like, oh, I want little Professor Perry to get it. Her real name's Dana, but we call her Professor Perry here. So her parents, I'm sure, knew she was going to be a professor. So they called her little Professor Perry. So they took her down to her friend's house, right? They gave her chicken pox so she'd hang out and get it over with and move on with it, right? And that was just kind of the way it was, right? That was not uncommon. You were not considered unethical as a parent if you did that. And, one of the things, though, that someone that has had chicken pox, someone that's had chicken pox, and they have a flare-up later in life, what does that present as? Shingles, right? Which is incredibly painful. And the thing that's really cool about kind of the herpes family of viruses is they actually hang out in neurons, right? So if you've known someone with shingles, they'll often get kind of like these, a lot of times it'll be like on their chest or their stomach, and they'll have like these tracks. And that's actually the virus moving along those neurons. And that's one of the reasons that it can be so painful. And for those of you that have had the chickenpox virus, one of the things that does not protect, or not the chickenpox vaccine does not protect you from, is actually getting shingles. All right. And so I think it's somewhere in your 50s, if you want, is when they start to offer up the shingles vaccine, right? But they usually don't give it to you until later in life, and unfortunately, some people get it before that, right? But again, it's a latent infection that kind of hangs out and then crops up, right? And so viruses, all these different cycles, and sometimes you'll even see kind of, I say unique kind of variations, of this. HIV is, I would actually kind of classify it as almost a little bit of an in-betweener. It's kind of a cool combination of a chronic and latent infection, but we'll talk about that a little bit more. So I do want you to kind of know the basic premise of what's going on in these different type of infections, right? And it's just important for those of you that are going into healthcare, right, to realize just because you're not showing symptoms, right, does not mean that you're not infectious, right? So yeah, I just totally took viroids out of here. Not that I don't think viroids are cool, but viroids primarily infect plants. We do think viroids might be present in us, but they're kind of hard to identify and we're not sure what they do yet. So let's talk about prions really briefly. Remember, if some of you want to do a prion disease, right, you totally can do that. But this is neuronal tissue from different prion-associated diseases. Here on the left, you can see normal tissue. Here is Kuru, right? This is caused or spread by the consumption of brain tissue from the deceased. This is classic Creutzfeldt-Jakob disease. And so if you were to eat meat infected with BSE, bovine spongiform encephalitis, mad cow disease, right? This is what you develop. There's also a genetic version of this. And this is a common prion disease in sheep populations. And I know some of you have not taken ANP, perhaps. I know some of you have. But spongiform, right? Think about a sponge. A sponge had a lot of holes in it, regardless of your ANP background, all right? This is neuronal tissue. This is neuronal tissue with holes, all right? Holes in your neuronal tissue are not a good thing. So there are a lot of severe symptoms that ultimately result in death if you develop a prion disease, right? So what's the basic premise of how a prion disease works? Well, so a prion, right? Specifically, we call it a prion protein, and we actually have two classifications. So we have the prion protein cellular. This is normal. We all have prion protein in our brain. Every single one of you, all right? But luckily, it's in this cellular form. And I admit I need to go and do this. We're still not quite sure, as of not that long ago, what these prion proteins are doing. The cellular one, right, this normal one, we think they might be important for some type of transport or signaling. Regardless, right, they're part of your cell membrane, so they're there, all right, and we think they're doing something with signaling or membrane transport. The important thing about them is they're easily degraded. So within your cells, you have something called proteases, and proteases break down proteins, potentially if you're using it as an energy source, but they're also going to be breaking down proteins as your cells remodel. Those of you that have had an ANP1, right, we talked about osteoclast and osteoblast, and how every year a large percentage of your skeleton is essentially rebuilt. That's normal, right? You want to remodel. You want to make sure things are working properly, that they're new and not broken. And so proteases help you break down and then build new stuff as you remodel a cell. And that's totally fine. What becomes an issue is when we have something called prion protein scrappy, all right? And the scrappy version, right? Remember, that's what we had the prion disease in sheep, right? This is not Scrappy Doo. Is he Scooby's cousin? I think, is that right? Does anybody know? Is Scrappy Doo Scooby's cousin?
Yes.
Right? Okay, good. Right, so please don't get those confused, right? But in this case, that scrappy version of the prion protein is severely misfolded, all right? And I'll show you on the next slide. We're not entirely sure why this happens, but when it becomes severely misfolded, it can no longer, all right, be broken down by proteases, all right? And when it can no longer, oh, go ahead.
Sorry, I was reading through the textbook and it said only like 40% of the population is susceptible to prion diseases. Do you know why?
Ooh, I do not know why. And I'd have to go back and look in the textbook because I'm not as familiar with this version of the textbook. Did it say they're susceptible to spontaneous formation of the disease, or are they saying that I could ingest a prion protein? Did they specify there? Do you remember?
I don't remember if they did.
Okay, so that would be really interesting to look at. So I'll show you here on the next slide why it's such an issue. I'll have to leaf through your textbook and see if I can find that, or go on PubMed and see if I can find some new papers where they discuss that, because that's interesting. I hadn't heard that. But anyway, right, so the protein's severely misfolded. When it's misfolded, we can't break it down. And so it starts to aggregate and kind of clump up. And so it's different, but in a way kind of similar. If you've known someone that has like Alzheimer's, and maybe you've heard of like Lewy bodies, right? And so Lewy bodies are basically these protein aggregates that the body can't break down, or that can't be broken down by proteases. And so when you have these aggregates, the body is trying to remove them, right? It's trying to get rid of them. And in that process of trying to remove them or get rid of them, that's what's leading to the formation of those holes in neuronal tissue, all right? And so the huge issue is that this is kind of a sick, twisted game of tag. And so when you encounter the scrappy version of a prion protein, and it touches or interacts with a cellular version of the prion protein, it causes that once normal version to misfold, all right? And so this is why the disease is deadly, all right? Because it's essentially a game of tag that doesn't stop, all right? Because then my little friend here, right, becomes a misfolded prion protein, and he can then interact, right? And it just keeps on going and going. So that's why I need to look, because Megan's point's really interesting. I'm not quite sure what would make someone resistant. I guess, I don't know if a prion protein cellular could somehow avoid that misfolding. That's interesting. I'll have to go and check that. But basically, this is happening a couple different ways, right? And so in some cases, right, there are spontaneous mutations, right? It's not necessarily something you interact with. A mistake is made, and that mistake that's made eventually ends in a severely misfolded protein. All right. The other link, and it's not really put here, is that we do know there is a genetic component. So there are some prion diseases that have a genetic link to them. And again, for whatever reason, that genetic link increases the likelihood of one of those prion proteins, the cellular version, misfolding. And once it's misfolded, right, it then interacts with other normal proteins and causes a misfolding event. And then we say inoculation, right? This would be the equivalent of if I said, right, you were consuming beef from a cow that had mad cow disease, right? And one of the things that I don't really have here on the slide is that these are incredibly resistant. These proteins are incredibly resistant to heat and incredibly resistant to chemicals that we typically use to disinfect labs. And so in some cases, this makes research really challenging because it's very expensive. The amount of money, so a lab that does prion research, that lab space essentially can then only be used for that type of research. Because it is almost impossible to sterilize or clean instruments that have been used in prion protein research. Pretty much the majority of the stuff that's used has to be disposable, which is also incredibly expensive, right? And is using a lot of resources. And so it's a very tricky thing to do research on, and there's no known treatment, right? They don't respond like viruses. They don't respond like bacteria because it's just protein, right? It doesn't have a nucleus. It doesn't have a membrane. It doesn't have genetic material that you can attack. It is a misfolded protein. So, transmissible spongiform encephalopathies, right? So prion diseases in animals and humans. So I mentioned scrappy, right, in sheep and goats. BSE is in cattle. So again, this was the mad cow disease issue. And I think I mentioned this was spread so easily because you had some cows that had BSE, and you can go and look online and find videos of cows with BSE. It's very evident in the late stages of the infection that they have something serious going on. So part of the problems was too, farmers didn't want to lose money, and so they would still slaughter these animals for meat. But the most important thing that I mentioned was after slaughter, right, you had a bunch of leftover cow parts. Those would be ground up and then fed back to the animals as feed. And so it would spread through herds that way. You have prion diseases that affect mink, deer, moose, cats, ungulates, ostriches. And so it's present in a lot of different animals. I think I still have the paper in here. There's chronic wasting disease that isn't infecting some of our deer populations, and it's actually a prion disease. I haven't seen anything yet, but I haven't really searched that deeply into transmission of prion diseases from deer to humans, right? Because sometimes we hunt deer. Kuru, I mentioned, right? Consuming human brains from the deceased as part of the burial ceremony. And then you have variant Creutzfeldt-Jakob disease. I'm sorry, this isn't the classic, is the genetic one. The variant one is if you were to eat or consume beef from a cow that had BSE or mad cow disease, then you would develop basically the human version of mad cow disease, right? So here's some of the different animals, so be careful there. Inherited, right, the genetic component. So this is the classic Creutzfeldt-Jakob disease, fatal familial insomnia, Gertzmann, Straussler, Schenker. I mean, that's a mouthful here, but these are prion diseases that have a genetic component and typically kind of start to appear a little bit later in life, right? So it can be quite shocking. Yeah, and this is just a paper, now it's 2013, but it's chronic wasting disease. And so it's just talking about the presence of these prions in the soil, potentially in deer populations. And so this is not a huge problem here, but it's something that we are concerned about and aware of. All right. So I want to